Hi, everyone. I'm Jeff Erlich, Assistant Professor of Neuroscience at NYU Shanghai. And my lab studies behavioral neuroscience. And today, I'm going to tell you about a project about the neurobiology of poverty. And I titled the project Towards a Neurobiology of Poverty because this is a difficult question, and it's really the beginning of the project. So I won't have any really satisfying answers for you. But I hope that our approach to the question you'll find it interesting. That is, in countries in Africa, people die at 55 on average, whereas in the United States, people live to over 80. And of course, poverty has many causes: war, famine, epidemic. But even within the United States, there's over a 20-year gap in life expectancy between the poorest and the richest areas. And one of the most tragic things about poverty is that it's passed from one generation to the next. Children born into poverty have less access to food, less access to healthcare and education. This leads to missed opportunities and stress, and this leads to lower income, and then their children share the same fate. So what does this have to do with neuroscience? Poverty is normally thought of as a social problem that's dealt through policy and social science. Well, there's really two dimensions of poverty that link it to neuroscience. One is that poverty is a state of chronic stress, and the second is that poverty involves financial decision-making. And these are both things that neuroscience has studied relatively extensively, especially stress. So the first thing is that poverty is a state of chronic stress, and this can be shown physiologically. If you measure the blood pressure or hormones of people in poverty, they're highly elevated compared to normal population, and this is true even in children living in poverty. So it's important to show this in children because with adults, you could argue that their bad life choices, their smoking, their drinking, their lack of exercise, is what led them to have high blood pressure. But it's hard to make the same argument for children. And neuroscience has been studying stress for over 100 years. And of course, we all use the word stress. You know, I'm stressed about work, I'm stressed about giving this talk, I'm stressed about this or that. But in biology, we have a more specific definition of stress. And a stressor, something that gives us stress, is anything that takes us out of balance. And we use this word homeostasis, but it's really just a sense of balance with the environment. So hunger, thirst, being tired, being lonely, feeling out of control, these are all stressors. And normally, stress is actually good, because let's say I'm hungry, so I feel stress. The hunger is motivating me to do something. It's motivating me to go out and get food, and then I eat, and then I bring myself back into balance. But if I'm living in poverty and I can't afford to get food, then my hunger persists, and that acute stress, which made me seek food, actually leads to chronic stress, which creates long problems. And for people sitting in the audience here, I don't imagine most of you are suffering from hunger, but we all have stress related to, let's say, work deadlines. And if, let's say, we have a deadline, we have to hand in some work, and that makes us work hard, and we do well. So that was good stress. The stress made us do something, and we achieved balance again. But if we have too much work, and we're constantly missing deadlines, and we can't re-achieve balance, then that leads to chronic stress. And we know chronic stress has serious long-term negative consequences for the body and the brain. In the brain, chronic stress leads to weakening of the parts of the brain involved in memory and executive function, things like self-control and decision-making. And it strengthens the parts of the brain involved in habit and instinct. And this has been pretty well established. So this is the first link between poverty and neuroscience. The second link, as I said, was financial decision-making. Neuroscience has been studying decision-making for quite a while, and we have a relatively good understanding of the neural circuits involved in decisions, although definitely not a complete understanding. It's still a very active area of research, as is the effects of stress on the brain. So people in poverty tend to make poor financial decisions. They carry high-interest loans, they fail to save for retirement, etc. And there's been growing interest in how these poor financial decisions can contribute to the cycle of poverty. And a really important dimension of these financial decisions is what we call intertemporal preference. That is, how much I prefer my future self over my present self. So if I favor my future self, then I'm patient, or if I favor my present self, then I can be considered impulsive. Let's say I won the lottery today. My present self may want to just buy a fancy car, get a fancy apartment, but my future self doesn't want that. My future self wants me to save that money so I can have a comfortable retirement. And there's fairly good evidence that people in poverty tend to favor their present selves, they're present-focused. Of course, one might say, well, they can't help it. If you don't have money for food, how can you save for retirement? But lab studies have shown that even people, middle class and upper class, if they're placed under chronic stress, they also shift their choices to be more present-focused. So this suggests that there may be a core biological mechanism by which stress shifts our focus from future to present, and that this could contribute to the cycle of poverty. But this is almost impossible to resolve in humans. There's so many other factors. There's education and culture and what you learn from your parents and a million other factors, genetics, that can't really be controlled. And so that's why we need an animal model to really resolve this question of whether or not chronic stress does, in fact, shift preferences to be present-focused, and how. And my animal model of choice is the rodent, rats and mice. So rodents are mammals. That's one of the reasons I like them, because humans are mammals also. And since they're both mammals, they have relatively similar brain architecture. So here you can see a human brain and a rodent brain, and you can see the scale bar. So obviously, there's a big difference in the size, and there's some difference in the relative contribution of different brain areas, but overall, they share similar architecture. Another reason I choose rodents is that there's been 2 revolutions in neuroscience that have really made just a paradigm shift in the kind of progress we can make at studying neurobiology of decision-making. The first revolution, I think, is underappreciated, which is the computer revolution. We've heard about that a lot today. So changes in the cost of electronics and how much data we can store, how many sensors we can use to track behavior and track neural activity. We now can generate terabytes and even petabytes of data and then store it and analyze it for relatively inexpensively. This is just impossible 10 years ago. And the kind of technology that we heard about from earlier allows us to now record neural circuits at an unprecedented level. Using the same kind of technology that we use to make computer chips, we can make probes that when we put them into the brain, we can record 10,000 neurons at the same time while an animal is performing a behavior. And this is really essential because the brain is so complex that traditional tools of looking at one neuron at a time just don't give you a full picture of how to connect brain activity to behavior. The second revolution you've also heard about a little bit, and that's the genetic engineering revolution. Over the past 10, 15 years, we've just seen an exponential change in our ability to monitor and perturb neural circuits using viral vectors to insert proteins from other animals or synthetically generated proteins that allow us to control neural activity with millisecond precision. And using these tools, we can actually control neurons in a certain part of the brain. We can control neurons that project from one area to another. We can control neurons that express a specific biomarker, like a receptor for dopamine or serotonin. And we can even control neurons based on their pattern of activity. Researchers at MIT have actually used this tool to turn a positive memory into a negative memory in mice. And researchers at Princeton have used a similar technique to take a mouse and teleport him. Basically, observe the pattern of activity when a mouse is in one place, and then when he's in another place, they replay that pattern, and the mouse thinks he's in the original place. So it's really like science fiction right now. And these tools have allowed us in my lab to use cheap electronics to build a high-throughput training center where we're currently training about 100 animals per day. And one person, one technician, can train 100 animals each day. And this is important because if we want to study complex cognitive tasks, we need a lot of animals. It also helps us study individual differences. And this is another thing that's really been missing from neuroscience. So these new tools really allow us to link DNA to proteins to neural circuits into behavior. And these genetic engineering tools probably won't be used in humans for many, many years. They're just not that safe yet for human use. But what they will allow us to do is identify specific neurons involved in some behavior or some disease. And once we know which neurons are the key neurons involved, we can then use pharmacological approaches to find safe ways to manipulate those neurons, which could be directly used in humans. So getting back to our question, we want to understand how chronic stress influences financial decision-making, in particular this future versus present self trade-off. And we want to study in the rat, and we want to apply those results to the human. So that presents a challenge. Rats aren't humans, and they don't save for retirement, But it turns out that intertemporal choices have been studied extensively in the lab. There's thousands of papers on intertemporal choice from economics, from psychology, and neuroscience. And the way we normally study economic choice in the lab is we make an offer to people. Do you want $10 today or $15 in 30 days? So let's just have a raise of hands. Who wants $10 today? Who wants $15 in 30 days? More people chose B, which is good. If you chose A, do not invest your own money. Find a financial advisor. Because $15 in 30 days is a 600% per annum return on investment. You'll never get that in the stock market. Okay, so this is how we study this in humans. So how do we study this in the rat? This is a problem, right? Rats don't speak English. 他们也不说中文. And so we need to find some way to translate this task. And so rats, they learn from trial and error. They learn from experience. And the other thing is, so that's one gap in the way we can study animals and humans, this verbal to nonverbal gap. The second gap is this time horizon gap. In humans, we make decisions, we do make short time scale decisions all the time. Like I, for example, often will pay $2 to remove the ads from my APP so that I don't have to wait for 20 seconds or something. So that's a kind of an impatient, short time scale decision. But we also make these long time scale decisions about days, weeks, and months, years. And rats we can never ask a rat, like, are you willing to forgo some food now for some food in 2 weeks? He'll never associate the reward later on with their choice today. So we have these two gaps that we have to overcome. And fascinating to me and to our group, we, despite thousands of papers on this topic, no one had really answered this question of whether the kinds of decisions you study in the rat, that is, waiting for seconds for a reward based on experience, are these the same cognitive processes that humans use when they make verbal decisions about days or months or years? So we decided to answer that ourselves. So how did we approach this? Well, what we did is we decided to train the humans like the rats. So we brought them into the lab, and we basically made a video game for them. And we didn't tell them what the video game was. We just told them, every time you see coins, that's worth real money. And then we shaped them just the way we shaped the rats. We made very easy steps until they figured out what to do. And what we did is we trained them that a high-pitched sound, like ee, that meant a large reward. And a low-pitched sound, like ooh, that meant a bad reward, a low, small reward. And for delay, if there was a slow modulation, like wah, 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 that meant a long delay. And if they heard a fast modulation, wah, 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 that's a short delay. And so on every trial, they would hear a sound, and they would have a choice. Do I choose to wait for that offer, or do I take a small, immediate reward instead? In this graph, see the trade-off here. So if the pitch is high, that means the reward is large, and if the delay is short, then you're happy, right? You like large rewards with short delays. That's good. And you don't like small rewards with long delays. No one wants that. And so this might be like an average subject, but not all subjects are the same. A patient person might be willing to wait for most of the offers that we give them. And an impatient person might be unwilling to wait for almost all of the offers that we give them. So we bring people into the lab, and we train them on this nonverbal task, and we do it over three different sessions. It takes them a few hundred trials to learn the map between the sound and the offers. And then after we're done, we bring them back, and we give them the classic choices written in language. We have both English-speaking subjects and Chinese-speaking subjects, and so we, depending on the subject, will present in different languages. And then what we ask is, are the people who are most impulsive in the verbal task also the most impulsive in the nonverbal task? And what we found was that there was an extremely high correlation, actually a shockingly high correlation. So here what we see is that the most patient subjects in the verbal task were also the most patient subjects in the nonverbal task. And the most impulsive subjects, the least patient subjects in the verbal task, were also the least patient subjects in the nonverbal task. And this was really important because what it suggested to us is the difference in people's preferences when they're making decisions about now versus the future, whether it's based on experience or whether it's based on a verbal offer, they're really expressing the same preferences, which suggests that they're using the same cognitive strategy, which suggests to me that I can use a rodent to study that behavior. And so we've trained rodents on these tasks, as well as other financial tasks. And as we speak, my postdocs are in the lab, and they're stressing our animals, and we're measuring their preferences. But I don't have the results yet, so I'm sorry to say that that's where I'll end. But I hope you learned something about how we're tackling this challenging problem. Thank you.